Before going to discuss different type of plasmids, that is, what are different type of natural plasmids or artificially created plasmids, first we can briefly focus on the basic properties of the plasmids, and we will start with the interconversion of plasmid DNA. So naturally, plasmid mostly present in the form of circular DNA molecule. अगर तो DNA के दोनों strands जो हैं वो intact हैं, तो उस सूरत में इसको कहा जाता है covalently closed circle DNA और CCC DNA. If one strand it is intact and other it is having a gap, so under such circumstances the plasmid it is called as open circle DNA or OCDNA. So naturally, when we try to isolate plasmid from the bacterial cells, so it is present mostly in supercoiled form. So supercoiled means that if the double helix structure of the DNA, it again having a coil, that it coils itself, then it is called as supercoiling. So plasmid DNA that we isolate from bacteria, so mostly it is present in supercoil configuration. That is, its double helix, it is again, or itself it can have super helical structure. So there are different type of enzymes that can interconvert supercoiled DNA into covalently closed circle DNA, that is cccDNA, or open circle DNA, that is OCDNA. And this interconversion of supercoiled DNA into covalently closed circle DNA or open circle DNA, it is mediated by different type of enzymes. Briefly, I can explain that topoisomerases, they are also one of the class of enzymes that can unwind the DNA when the DNA replication it is going on. So in the direction of the movement of replication fork, the stress is created on the DNA, and if this stress it is not relieved, then the DNA replication is affected. So by creating artificial nicks, so topoisomerases can relieve such topological stresses on the DNA. So topoisomerase can also unwind the DNA temporarily. In addition to that, if the DNA it is again converted into covalently closed circle DNA and we want to convert it into supercoiled form, then we can use DNA gyrase. So it can again rewind the DNA, but maybe in opposite direction, that is, it can introduce negative supercoiling. So if we can if we want to create open circle DNA, then the DNA it can be treated with endonucleases so that it can cut one strand of the DNA under some situations, so that we can have open circle form of DNA. So it can be explained with the help of a diagram that how enzymatic interconversion of supercoiled DNA into cccDNA or OCDNA it is there. In this diagram, as I told you that if you attempt to isolate DNA, mostly it is present in supercoiled form here. So this supercoiling, it is there naturally, it is present in the bacteria. So it can be converted into covalently closed circle DNA or cccDNA when we treat it with enzymes like topoisomerases, so that relaxed or covalently closed circle DNA it is produced. If we again want to convert it into supercoiled form, then again it can be treated with another enzyme, that is DNA gyrase. So it will introduce negative supercoiling. And if we treat supercoiled DNA with endonucleases, then OCDNA or open circle DNA it is produced, because one strand it is not intact. So such type of DNA it is called as OCDNA or open circle DNA. OCDNA it can again be converted into ccDNA if we bridge this nick with the help of DNA ligase, so that it can again converted into ccDNA, or ccDNA again can be converted into OCDNA if we again treat it with another enzyme which is called as endonuclease. So this is the enzymatic conversion of supercoiled DNA into cccDNA or open circle or OCDNA.